Hello everybody, my name is Anyssa Mhaia, and this summer I had the wonderful opportunity of working with Vanderbilt University's Mass Spectrometry Research Center, where we focus on optimizing matrices for negative ion mode multi-IMS. So before we get into it, what is multi-IMS? So multi-IMS is a technique in which we analyze the molecular composition of tissues by spatially mapping their molecular distribution using mass spectra. So multi uses something called a matrix, which is a small organic molecule that is meant to facilitate ionization of large molecules while also absorbing energy from our incoming laser to protect our sample. If we repeat this process over and over again on our tissue, we can get something that will turn into an image. So on the right, we have a diagram of what multi-IMS actually looks like. On the bottom, we have a matrix-coated tissue sample. If we hit the sample with a laser beam at a specific spot, then we can get a mass spectrum for that specific location. Now, if we do it over and over again in a grid-like pattern, then we will get a bunch of different mass spectra, each corresponding to a specific location on our tissue. Then we can take the average of these spectra and represent them with a pixel, which is how we get something called a total ion image. So IMS is this way of combining mass spectra with this rapid surface sampling process in order to match each location of our sample to a specific ion. This summer, our goal was to develop a sample prep approach in order to create high resolution, so sub 5 micron images using MALDI. By being able to image at a very high resolution, we then would be able to turn it into a multimodal mixed matrix sample prep. And thus, we would then be able to apply different methods and modalities to the exact same tissue without having to get a new sample. So there are many different things that can affect the way that a signal is going to appear like. One of the main things is the type of matrix solution that is being used. This summer, we use three main ones. On the left is DMACA, in the middle is SuperBase, and on the right is Vandy 37. DMACA is a commercially available matrix. It works incredibly well in negative mode. However, we have not been able to use DMACA for high spatial resolution imaging because it tends to have gas phase reactions. In the middle is SuperBase. It is also another commercially available matrix and does do well in negative ion mode. However, it needs to be combined with another matrix in order to be successful as a matrix. On the right is Vandy 37. It is an in-house matrix developed here at Vanderbilt. It works very well in positive mode and actually has been able to image at a resolution below 5 micron, but it has only been able to do so in positive mode consistently. Especially with different types of tissues, we know that Vandy 37 does not do as well on human samples, for example, in negative mode. So this summer, our goal was to find the right ratio of Vandy 37 to SuperBase in order to maximize our negative ion mode signal. This is because we know that Vandy 37 by itself can already do high resolution imaging, so we were meant to focus on a negative mode improvement. So we tried different types of ratios, all the way from 1/100th all the way back to 1:1. And we found that a 1/4 dilution of SuperBase to Vandy 37 gave us the highest negative ion mode signal. We hypothesized that the primary matrix here, Vandy 37, would interact with SuperBase with a constant ratio. So no matter what concentration we keep Vandy 37 at, the ratio of Vandy 37 to SuperBase was going to remain the same, so a 1/4 ratio between the two. But before we can even get into optimizing our workflow to get a high resolution image, we had to learn what a regular multi-IMS workflow looks like. So for the first part of our project, I had to work with rat brains, since these are very easily found, cut at a 10 micron thickness. So using our cryostat, I cut the rat brain at this 10 micron thickness and then thaw mounted them onto ITO slides and washed them with ammonium formate. Then, because this wasn't high resolution imaging, I could use either DMACA for negative mode or Vandy 37 for positive mode as the matrix to be sublimated. And then to get the IMS data, we use a timsTOF flex at a 5 micron resolution setting. Then to get our images, we used Skills Lab to create our raw molecular images and combining our ion images. Then, if we want to further push our analysis, we can also do statistical analysis or pre-processing, which can be done to our IMS data. And so here is what the outcome of this workflow looks like. Here is a rat brain cerebellum at a 10 micron spatial resolution with a 5 micron laser setting. On the left is Vandy37, on the right is DMACA, and both of these were taken in positive ion mode. As expected, we can see that Vandy37 does outshine DMACA, since we know that Vandy37 works incredibly well in positive mode. Not only is the signal more clear and also more intense, but if we zoom in onto particular sections on the tissue, we can see that there are also some physical features that we would have missed of our tissue. The Purkinje cells that line the gray matter of the brain would not have been able to be viewed if we had just used DMACA in positive mode. It instead just looks like a purple blob almost. Now, this analysis can also be applied vice versa. If we had done these images in negative ion mode, we would have seen that DMACA outshines Vandy37 as well. And so after learning the general workflow for a MALDI IMS experiment, we then worked on bettering it to create a high-resolution MALDI IMS workflow. Since we were now being able to image at a high resolution, we were able to use more precious samples such as HuBMAP human kidneys, which these were cut at a 6-micron thickness using our cryostat. Then we thaw mounted them onto ITO slides and washed them with ammonium formate like before. The main change in our workflow was the way that we sublimated. Before, we sublimated either Vandy37 or DMACA. Here, we sublimated two matrices together. So we did a combination of Vandy37 with superbase at a 1/4 ratio and sublimated them simultaneously. Then to get our data, we used a timsTOF flex at a 3-micron spatial resolution setting. The rest of our experiment kind of went the same way as we did for a regular MALDI IMS experiment, where we created our raw molecular images using SkillsLab, and then if we wanted to, we can also do some pre-processing or statistical analysis on our IMS data. Now, given that we had this workflow, we wanted to make sure that it worked first. So as mentioned before, we knew that Vandy37 has and already did image at a 3-micron resolution. So before we even tested negative mode with our new matrix solution, we first ran positive mode with Vandy37. So here is a 3-micron positive mode IMS image. Above we have Vandy37, and below we have Vandy37 with a 1/4 dilution with superbase. As we can see, not only is the Vandy37 image a lot clearer, but the signal is also higher by a whole order of magnitude. If we zoom in onto our tissue samples, we can also see that we can see direct details of the human kidney, such as gloms and other parts of the tissue that we obviously cannot see with a Vandy37 dilution below. And so, given that this worked, we knew that we could then apply it to a negative mode signal, since we knew that if our 3-micron in positive mode IMS did not work with Vandy37, we would have messed up some sort of step in our sample prep. But given that it worked, we moved on to negative mode. And so here is our very first 3-micron negative ion mode image of the human kidney that we were able to do here at Vanderbilt. So not only is our image beautiful and we can see all the heterogeneous gloms and all of the details in our tissue, we can see that the signal intensity is also a whole order of magnitude higher than if we had used Vandy37 in negative mode. Now, as expected, we knew that Vandy37 does not work well in negative ion mode. However, what's interesting here is that we can see that when we choose in ion mode, we either have to sacrifice one or the other. So in positive mode, we can see that we will have to sacrifice negative mode and vice versa. Now, the fact that we were able to image at a 3-micron resolution also meant that we could now apply different modalities to the same tissue. So below we can see that we H&E stained the exact same tissue that we used to run for this particular image, and then we scanned on our Axio. As we can see, there are no marks. Our tissue does not look damaged, and we don't even see a burn pattern or a burn mark on our tissue. If we had used a lower resolution imaging, then we would have definitely seen some burn marks on our tissue. So being able to image at a 3-micron resolution means that we can now also apply different modalities to our tissue. And this is because we use a laser power that is a lot lower for 3-micron imaging. So in conclusion, we were able to push the limits of not only our sample prep, but also our commercially available mass spectrometers to image at a sub 5-micron resolution in both positive and negative mode. As a result of what we were able to find, we were able to get the best of both worlds for MALDI IMS. We were able to image at a high resolution while also keeping our signal intensity quite high. Being able to image at a 3 micron resolution now opened the door for us to create a multimodal mixed matrix sample preparation. And by imaging at this high resolution, we are thus not damaging our tissues as much. And so we can then apply different modalities such as a stain or immunofluorescence to be able to reveal more information than we would be able to get from just regular MALDI IMS. In the future, we're hoping to apply this sample prep to different types of tissues, possibly other HuBMAP tissues or other parts of the brain, such as on the right, we have an image of a rat brain hippocampus. I would like to give a big thank you to Martin Dufresne for being my mentor for this project. I learned so much from all of his help, and to my PI, Jeffrey Spraggins, for all of his support throughout this summer, and to the VU MSRC members for their help throughout this internship. I'd also like to give a big thank you to the NIH and HuBMAP for this wonderful opportunity that I would not have been able to do without them. Thank you so much.